My name is Sanjay Vignesh, and this is my presentation on the therapeutic analysis of Plerixafor on relapsed acute myeloid leukemia patients, and this is for the Polygence Symposium of Rising Scholars of Fall 2024. So first off, who am I? Like I said, my name is Sanjay Vignesh, and I am currently a sophomore at Doherty Valley High School, and some of my hobbies include robotics, aerobotics, and biomed. And this is a picture of our robot for this season on the left. And the mentor who guided me through my project is Jen F, and my showcasing mentor is Janice H. And I chose to research therapeutics because even though a lot of my hobbies are like geared towards engineering, I still wanted to experience the biotech field so that I have a better understanding of my interests. So that's why I decided to do research on therapeutics. So first up is the disease I am researching, and that is acute myeloid leukemia. So what is acute myeloid leukemia? So acute myeloid leukemia, commonly referred to as AML, is a hematologic cancer, which means it is a blood cancer that starts off in the cells that will develop into different types of blood cells, including white blood cells, red blood cells, and platelets. In AML, the acute means immature cells spread quickly, and the myeloid part of the name means that it develops in the monocytes, and it affects almost 20,000 new patients yearly. So how about the origin of AML? So AML originally, it comes from when the bone marrow DNA is mutated to make bone marrow cells divide. And because these cells divide, they like multiply and grow, which is called myoblasts. But pretty much when they build up and grow, it just crowd outs and interferes with the healthy cells. So now we're going into stem cell niches and how they affect my project. So stem cell niches are microenvironments in a specific part of the body where stem cells reside and receive informations and signals. So the specific stem cell niche that I'm looking into is the hematopoietic stem cell niche, which from the name you can imply that it moderates hematopoietic stem cells. And the special case it has in this project is that it actually provides protection to the LSCs, which are leukemia stem cells, and keeps them dormant, which can cause AML relapse during chemotherapy. So now onto the chemotherapy slash relapse part of the presentation. Chemotherapy is used as a form of treatment for AML, but it promotes relapse in chemo-resistant LSCs, which can be protected by the stem cell niche. So, to conjure this problem, scientists have discovered various ways of dealing with LSCs, either being LSC eradication through anchoring interference or mobilization of LSCs out of the hematopoietic stem cell niche. So if they are still dormant in the stem cell niche, the surviving LSCs can diversify and result in more aggressive forms of AML, which is known as AML relapse. So the specific drug or solution that I'm looking into today is Plerixafor, which is specifically a CXCR4 antagonist. And scientists are able to use Plerixafor in combination with other chemotherapeutic medications that I'll go over to disrupt the interaction between the chemokine CXCL12 and its receptor CXCR4. This interaction mobilizes LSCs out of the protective niche, which makes them vulnerable to forms of treatment, and this strategy can be used to eradicate dormant LSCs, which are the root cause of AML relapse. So the main study that my paper goes over is a phase 1 clinical trial conducted by Gail J. Robaz. And in this study, scientists combined plerixafor with cytarabine and daunorubicin to test the safety and tolerability of plerixafor with mobilizing LSCs in newly diagnosed AML patients. The results of the study, they yielded a 67% complete remission rate, which suggests that the mobilization increased LSC's vulnerability to chemotherapy, which possibly improves treatment outcomes for patients with relapsed AML. The benefits of the study can include sensitizing LSCs to chemotherapy, which is a step in the right direction for treating relapsed AML patients. So then there's a second study that we go over, and in this study, it was conducted by Jeffrey L. Oy, and the scientists tested plerixafor for its potential to induce sensitization of LSCs to decitabine. And the scientists conducting the trial, they actually hypothesized that combining the plerixafor with the decitabine would improve treatment outcomes through the mobilization of LSCs. The results of this study had a response rate or a complete remission rate of 43% and an overall survival of 11 months over the course of a 4 to 6 month long treatment. And the specific benefits of the study include using combination of plerixafor and decitabine as post-remission therapy for older patients with AML to improve treatment outcomes through the mobilization of LSCs and the alteration of the pharmacodynamics of decitabine. And this is the last clinical study that we're analyzing, and it's a phase 1/2 clinical study also conducted by Gail J. Robaz. And in this study, the scientists used Plerixafor to treat relapsed AML patients in combination with mitoxantrone, etoposide, and cytarabine. So the results of this study yielded a 39% complete remission rate and a 2.5 times mobilization of AML blasts with no preferential mobilization over healthy cells. The specific benefits of this particular study include enhancing the cytotoxic effect of chemotherapy for relapsed patients by disrupting by disrupting the interaction between AML blasts and the marrow microenvironment with Plerixafor. So now we can go over some key study similarities and differences. So quite obviously, all three studies use Plerixafor in combination with other chemotherapeutic drugs to help treat relapsed AML patients. Um, all the studies also had around the same amount of participants, ranging from about 36 to 71 participants, and this means all the results were fair game. They weren't like they shouldn't skew the results. And another difference is that the main study achieved a 67% complete remission, while the other studies gained 39% complete remission with a 2.5 times mobilization of AML blasts and a 43% response rate with 11 months of survival, respectively. Now, a big difference that we have is that the phase 1/2 clinical study, the last one, is very distinguishable from the other two trials because one of the inclusion criteria is that it requires at least one round of induction therapy from the patients, while the other trials actually, which is very interesting, they exclude patients if they've had any chemotherapy for a prior malignancy within a certain range of time. It's possible that this could have skewed the results because we are testing for chemotherapy effectiveness. And one last difference is that the first trial was actually conducted in newly diagnosed AML patients, which I believe is a 20% myoblast rate. And the second trial was conducted on elderly patients. All of the ages are 60 and above. And then the last one is conducted on just AML patients, or sorry, relapse AML patients with, again, 20% myoblast rate. So, in conclusion, AML is a fast-growing hematologic cancer that develops and spreads quickly in the bone marrow, the bone marrow's myeloid cells. Scientists are able to use Plerixafor to disrupt the interaction between the CXCL12 and its receptor CXCR4, which mobilizes LSCs out of the protective niche and makes them vulnerable to chemotherapy. The future directions of AML treatment should lean towards personalized therapies using combinations of mutation-targeted agents aiming at LSC eradication with reduced side effects and non-mutation targeted novel agents directed to LSC eradication through interference with their anchoring to the stem cell niche. And according to this graph, the continued study of AML, the continued study of AML will result in increased patient survival and remission rates for over 20,000 patients yearly. And these are just some of the citations that I used in this project. And that's the end. Thank you for listening to my presentation.
Thank you so much. I mean, you had said in the beginning you were like really interested in robotics. What made you go down this road to do research on these cancer cells?
Well, I always had like an affinity for bio as well, and I just wanted to like I didn't really want to set my like major in stone yet without like having giving it a real chance. So I just wanted to give like the biotechnology field a chance by doing some research on it. Maybe I could get some experience in it. And I also did a summer program on therapeutics, just like to really be sure to lock in which if I want to choose engineering, if I want to go down biotechnology.
So have you decided?
Yeah, I'm pretty sure I'm going down engineering.
Yeah. I mean, it was a great presentation. So you're saying that that 67% had full remission rates
For the first study, yeah, for the main results.
from the main results. Did they talk about why the other ones did not have remission? Was there any type of risk factors or commonalities between them?
The studies didn't say specifically because they weren't like synonymous with each other. But if I had to make like an analysis, I would probably say it's because they use different types of drugs with each other. For example, the first one used daunorubicin and cytarabine, while the others use mitoxantrone, etoposide, cytarabine, and I believe the last one was the decitabine. So obviously that's like, a key factor because when I was in like actually when I did the summer program on therapeutics, obviously like different drugs have different like structures and functions, right? So I think that would like definitely affect the efficiency.
And did you find anything in the data that says that 67% is a good remission rate for that type of cancer?
I haven't found like a specific source, but 67% is generally like I remember when I was talking to my mentor, and I believe she was also a bio person. She said 67 is generally a pretty high number. So I would
Wow.
say that's a pretty nice success.
Yeah, that's great. I appreciate it. Thank you so much. It was a great lesson. And it's something I didn't really know much about, so I was well informed. Thank you so much, Sanjay.